So what if the cure to cancer isn't a drug, but instead a cell already inside your body? These cells are T cells, also known as T lymphocytes, a crucial part of the immune system that can be extracted from a patient's body, genetically modified, and programmed to target cancer. This approach is known as CAR T-cell therapy, and its success lies in the involvement of the patient's own immune system. But first, what are T cells? T cells are a type of white blood cell responsible for identifying and destroying infected or abnormal cells in the body. They recognize antigens, foreign proteins, and coordinate targeted immune responses. CAR T-cell therapy was first approved by the FDA in 2017 to treat children with acute lymphoblastic leukemia, a fast-growing cancer of immature white blood cells, particularly affecting the bone marrow and blood. Because this therapy is tailored to each individual, the first step involves making the treatment. Blood is extracted from the patient, and the T cells are isolated and sent to specialized labs where they're genetically engineered. T cells naturally have receptors, proteins on their surface, and during the genetic engineering process, a new receptor is introduced. This is called a chimeric antigen receptor, or CAR. These receptors are specific to proteins on the surface of cancer cells, allowing the T cells to bind directly to them. Once the engineered CAR T cell has the correct targeting receptor, millions of copies are produced in the lab. These are then returned to the hospital and administered to the patient through a single infusion. Clinical trials have tested this therapy on patients with advanced follicular lymphoma, a slow-growing but hard-to-cure type of non-Hodgkin lymphoma that originates in B cells. The results showed that the cancer was eliminated in around 80% of patients, and in many of those individuals, the disease didn't return even after 3 years. Another trial involving large B-cell lymphoma found that more than 30% of patients were still alive and cancer-free 5 years after treatment. So, why might this therapy be dangerous? CAR T-cell therapies primarily target mutated B cells, but many healthy B cells also carry the same surface protein, CD19. This means that CAR T-cells can't distinguish between cancerous and healthy B cells, leading to off-target effects. The destruction of healthy B cells weakens the immune system, making patients more vulnerable to infections. To manage this, doctors may use immunoglobulin therapy, replacing the antibodies normally produced by B cells, along with close medical monitoring. A second serious side effect is cytokine release syndrome, or CRS. When CAR T cells attack cancer cells, they release large amounts of cytokines, chemical messengers that regulate the immune system. But too many cytokines can cause an excessive immune response. CRS can lead to dangerously high fevers, low blood pressure, difficulty breathing, and in severe cases, can be life-threatening. The third major side effect is neurotoxicity, also known as ICANS, immune effector cell-associated neurotoxicity syndrome. Elevated cytokine levels can trigger inflammation in the nervous system, leading to confusion, slurred speech, motor dysfunction, and even seizures.